Thank you, Anna. Good afternoon, everyone. My name is Liz. So my formal title is Associate Director of Product Marketing. Long time ago, I was a bioinformatics scientist starting at PacBio working on basically our long-read RNA-seq algorithm development and later transition to product marketing. So these days, I talk mostly about work that's been done through my collaborators. I do not claim to have written the code or the figures that generated this talk. But the reason I want to start this talk here is that most of us who work in algorithm development is very focused on a very specific problem, such as how can I correct the splice junctions? How can I do a better prediction of RNA or, let's say, Hi-C? But you want to zoom out to this picture, and I'll come back to this at the very end, is ultimately we're looking at genomes, be it human genomes or otherwise. Then we look at, from there, RNA is transcribed, and then it turns into proteins, and even proteins have modifications, proteoforms, and a lot of this dysfunction in the genome permeates into the RNA, then into the proteins, which causes disease. A lot of the work we do ultimately is to serve the understanding of basic biology, but to also understand how these dysfunctions cause disease so that we may correct it. I've been at PacBio for almost 13 years now, and we've started the HiTSeq method, which is PacBio's application of full-length RNA sequencing almost more than 10 years ago. The early HiTSeq papers, as you can see from the titles, mostly focused on de novo isoform identification. It wasn't because PacBio wasn't quantitative, but because there's only so much when you only have 1 or 2 million reads per run. But throughout the years, we've really increased our throughput. And one of the really game changer for us is actually this very cool molecular trick, is concatenating amplicons to larger fragment sizes that can be sequenced on PacBio. PacBio is a long-read sequencer. We work best with reads that are 15 to 20 kilobases. But if you look at your average 10x cDNA, which if you haven't seen before, has an average read length of 600 bases to 1,000 bases. If you look at your average bulk cDNA that's synthesized from commercial products, it's usually somewhere between 1 to 2 kilobases up to about 10 or 12. It's very hard to get really long transcript cDNA beyond that, mostly because of the limitation of the library preparation, not because nature does not produce long transcript. It totally does. But with this concatenation method, we are now able to actually increase the throughput of PacBio artificially beyond the capacity of the number of sequencing wells we have. This is the only marketing quote-unquote product page that I'm going to show, is that these are the 3 kits that we've developed to serve different concatenation factors. We sell 3 different kits that do 8-fold, 12-fold, or 6-fold concatenation. So based on different sequencers, you can actually get up to 120 million reads per run. The rest of this is really musings from a PacBio RNA product perspective. Is the questions I get asked when I try to go out and tell customers that, hey, you should use long reads for RNA. The first thing they always ask me is, okay, cool, you tell me how many reads I should sequence. The really answer is only, it really depends, because what are you sequencing? If you're sequencing the liver, if you're sequencing cancer samples, it varies. But nevertheless, I had to go out and find some data to show the trends. We have sequenced many cell lines, and this is a collaboration on patient samples of heart and cerebellum, where we plotted the saturation curve on the left-hand side, showing the number of known isoforms. It's easy to look. Novels are going to be much more difficult to quantify, so we only use the GenCo known isoforms, and say, at what point do we start to hit a saturation point of known isoforms? We've plotted this for many different samples, and over the years, my conclusion is at around between 10 to 20 million reads, you're going to start hitting diminishing returns for isoform identification. The other way to look at this question is to look at the right-hand side, which is, how much more sequencing do I need to find something new? The discovery curve. At around 10 to 20 million reads, you have to sequence another 1,000 reads to get one more new isoform. I know most of us are bioinformaticians, but your PI is paying for that sequencing. And at some point, they're going to say, am I hitting diminishing return for finding something that is useful or actionable? The next question that we really had, and is the bulk of this talk, is thinking about, now that we have a lot more reads, can we definitively say that we can quantify with long reads, especially PacBio Connect's data? And also questions that most of you do not have to think about, but I do, which is how replicable is this data from run to run? If I sequence it on Monday and Thursday, if I use this SMRT cell versus that SMRT cell, if I buy this kit from this month and next month, how different it's going to be? What biases exist because of the library preparations and the sequencing that exists in PacBio systems? And finally, the big question, if you've been sequencing on Illumina short reads all your life and you come to use long reads, what's the comparable read depth? To answer these questions, we collaborated with Mark Robinson's lab at ETH Zurich and Gloria Schinkelman's lab at University of Virginia. They had access to the WTC11 cell lines. They grid over 5 different days, differentiated into endothelial cells. And we also added SIRS spike-ins. SIRS are long RNA control spike-ins. So there's a ground truth in there. We also generated replicates over multiple days. We generated companion, long read, and Illumina short read data. Just to show you the scale of the data, the Illumina data was over 11 different samples, so 5 different time points. Some of the days had replicates to a total of over 600 million reads. For the PacBio Kinetics data, we generate somewhere around 500 to 600 million reads. But one of the things that you have to notice when you look at just the reads is not a sufficient quantifier of the data difference, because if you look at the read lengths, of course the kinetics, after deconcatenating, remember I said they're sticking different amplicons together, well, these are back to the original cDNA sizes. They have a mean around somewhere between 1 to 2 kb. That's what the pink violin plot is. And the Illumina data is all going to be exactly the same. Why? Because these are 2 by 150 base pair reads. Then if you look at that, given that the read depth is relatively comparable between short and long reads, if we then extrapolate with the number of read length differences, the number of bases in Kinetics is actually 10 times more, simply because every Kinetics read is going to range somewhere between a few hundred bases up to 10 kilobases. Then let's look at quality. So I know some of the talks today were very interested in correcting some of the splicing errors and other technologies. One of the things I think is less of a problem in PacBio data is accuracy. I'm not saying it's 100% accurate. As you can see, it's not. But there are going to be remaining sequencing errors, but also variations, because we estimated the alignment quality based on HG38, but the WTC cell line is not HG38 reference. So there's going to be some of natural variation. Based on the alignment, we estimated that the average Kinetics data is 99.7% accurate, whereas Illumina is 99.4% accurate. The next question we were interested in is reproducibility. Because we have different days and replicates, we're able to actually look at the Pearson correlation of different day replicates. Sorry, same day, different replicates. As it turns out, on this end, both Illumina Kinetics do incredibly well. The correlations are very high for the same day samples. Just to show that this reproducibility and consistency is not just on this data set, we've actually showed this on internal UHR samples run on Revio, actually across different chemistries. And then we also ran it on another long-read sequencer that we have called the Vega. And on the left-hand side is the average read length of the cDNA that's generated. Again, it starts around 1 to 2 kb and goes all the way up to 10 kb. And there really is not a difference between the different sequencers or chemistries. We also looked at the gene and isoform abundances across the different platforms, and they're relatively consistent as well. We then looked at some of the unique characteristics of the fact that we have this time point into endothelial cell differentiation. Both Illumina and Kinetics data can easily identify the differentially expressed genes that are markers for endothelial cell differentiation. In fact, because these gene expression differences are so strong because it's cell differentiation, that if you downsample the reads down to 5 million reads, you can still identify most of these DE genes. And that's what the upset plot is showing here. Now we're getting into the area where there is a difference between short and long reads. The first is people have chosen to use long reads because it's easier to detect full-length isoforms. Less assembly, or should I say no assembly, is required. The first is using the synthetic SIRS spike-ins that we've added into the WTC11 cell line. We're able to calculate the precision and recall for detection of these SIRS. Kinetics outperforms Illumina in terms of both precision and recall. We then also use that to look at the rest of the WTC11, the actual biological samples, and identify the novel isoforms that exist in these samples. I will say the upper right figure is not produced by me because the x-axis and the y-axis is horrible for the left and the right. CPM stands for counts per million. If you look at the low abundance counts, so 0 to 10, you could see there are thousands of novel isoforms in the NIC and NNC category, but still coding. So these are novel isoforms that very likely produce novel proteins, but they're expressed at low amounts. There are, however, some novel isoforms, the CPM greater than 10, if you notice the y-axis, like I said, these figures should not have been stacked next to each other because they look almost the same. But the right one, CPM greater than 10, has an y-axis of only around 600. So we do see novel isoforms that are highly abundant, but there are much fewer of them. Most of the novel isoforms that you'll be sequencing are lowly expressed. We then also benchmark different quantification tools. So this collaboration, the goal wasn't to create a new isoform detection tool or create a new isoform quantification tool. Many long-read tools have been developed for both of these purposes. We benchmarked a few of them: Bamboo, LR Callisto, IsoQuant, and Orfish. And for the Illumina data, we used Salmon. What we found was that in terms of runtime, Bamboo and LR Callisto or Orfish are all really fast. In terms of memory usage, Bamboo and Orfish also do really well. And then we use serves, again, as a control to calculate the false discovery rate and the true positive rate. Here you can see that Orfish and Bamboo doesn't do as well. However, that is because serves are only 78 isoforms, and they're synthetic. They're artificially made, and they were made by people who have very sick minds because they designed a serve 301 and 303, where if you look at the junctions, it has exact 303 is a perfect subset of 301. It uses exactly the same first two intron chains as 301, but 301 has two additional 3' exons. So this is a good example because nature does also produce something like this. Definitely. And I think for algorithm developers, it's important to think about, can I actually differentiate these two without making the assumption that if something is a subset of the other larger isoform, then therefore the subset must be just a library artifact and should be automatically merged into the longer data set. The next thing we looked at is the difference in the ability to quantify transcripts between short and long reads. And here are some of the interesting observations by my collaborators is looking at how inferential variability affects short read quantification. They named two phenomena that they observed that is more plaguing short reads than long reads. One is called transcript flips. And in this case, if you look at the three replicates of the same day, there are 4 isoforms in this gene. And I'm showing in blue, just as an example, one of the isoforms. If you look at that isoform over the 3 days, in the short read based quantification, the first day shows that it has some amount of quantification, but then on day on the replicate 2 and 3, it's completely 0. And why is that? What happens is that the collaborators named this the transcript flip phenomenon, where when short read isn't able to definitively assign a junction or a subset of reads to multiple isoforms, as you see these 4 isoforms share, especially the second and the third isoform, share so much similarity, what will happen is they will start randomly assign the expression of the transcripts across replicates, not knowing that we ran these on the different replicates independently. So it doesn't know the 3 replicates are actually associated. So it's going to randomly assign the expression to these very similar isoforms. The other phenomenon that they observe is the opposite of transcript flip, where like the same transcripts have very either near 0 or super high abundance over the replicates, is actually when you don't know what the answer is based on short reads, you might as well equally divide them, because the optimization or the scoring function you use probably would do just as well if you say, hey, given this junction is shared among all isoforms, I might as well say there's 25% equal chance that they all express, right? So here's an example. Here's a gene with, again, 4 isoforms. The gen code is shown in green. And then if you look at the short read data, every little bit is a short read junction. Then you have the long read data, pink, show predominantly one isoform, and that is the one that's shown in blue. So then if you first look at the pink bars on the right, Kinect's data will quantify these as all being really well expressed for this isoform. But if you look at the other isoforms, the other pink bars, they're very low. That's what the data is showing. There's very few expression of the other 3 isoforms. There's only one dominant isoform expressed. But then if you look at the short read data, it's showing all 4 isoforms as being equally expressed over the replicates. When you look visually at the UCSC browser, it does not look like the short read should be supporting equal expression of these 4 isoforms. But probabilistically, it probably looked just as good. With these observations, my collaborators corrected for inferential variability and said, what would the quantification look like between the short and long reads if I removed every gene and transcript that has high inferential variability and put the 2 platforms on the level playing field? When you remove anything that's hard for short reads to quantify, then short reads actually quantify similarly to long reads, both on the absolute and relative quantification for both genes and isoforms. The last thing I want to show is, to be fully transparent, is there are biases with long-read cDNA data, especially cDNA, because of the way cDNA is synthesized. This is not the first or the last figure that's going to show when you do long-read cDNA, there's going to be a 3' bias because of the way libraries prepared. Most library preparations start with the 3' end priming on the DT primer and then goes back to the 5' end with template switching. The 3' end is therefore going to be reliable because of the priming on the poly A tail, but the 5' is not going to be reliable for 2 reasons. A, RNA degrades on the 5' end, and second, depending on what enzymes you're using for producing that cDNA, it might not get to the very 5' end of that molecule before it falls off and goes back again. This is not a problem with short reads because you're fragmenting the RNA and then you're sequencing pieces of it. So you're going to get better uniform coverage over the gene body simply because of the way the cDNA is prepared. The other thing is also because of the way we do the library preparation, which uses some beads, this is molecular biology hour here, it's going to be biased towards slightly longer transcripts. And they plotted here that Kinect actually has a slight bias towards over 1 kb transcripts. And that means if you have a 100 or 200 base RNA, it's going to be less detected with long-read data. So a brief summary of what I've said so far is that we've shown that there's strong quantitative support. Long-read will avoid more inferential variability problem and actually detected more differentially expressed isoform events than short reads. We've shown that both platforms have really good replicability. There's some bias in long reads with this 3' end and longer transcripts. But what we weren't able to show in this paper that we had vigorous debates on, and I'd love to have some of you think about this, is what is a comparable read depth between short and long reads? And I'll tell you why we weren't able to even tackle this problem, because we were not able to agree upon what is the equivalence of a short read detected isoform versus a long read detected isoform. And I'll show you some of that evidence later. I'm going to switch gears quickly and go really big picture, which is to show that there's many, many long-read RNA-seq data that's been developed by the community, and you've heard some of the talks today about it. But as someone at PacBio who's looking at the entire product, I'm not interested in just one aspect of the RNA analysis. I'm interested in everything from base calling to primer trimming to getting the poly A tail right to secondary analysis to visualization and differential expression tools. In the next few slides, how much time do I have left? I'm over time? Okay. Then you know what? I'm going to skip all of this then, because I don't want to get into our keynote speaker. So I'm going to go, skip all of these, and just show some of the last thing I want to talk about. What's coming in the future for long-read bioinformatics? I really plead you to think about scalability. Some of the questions I asked in the audience today is, can you process 500 million reads in less than a few minutes? Or actually, all right, let's put it this way, 100 million reads in a few hours and a billion reads or several billion reads in a week. Because if your algorithm cannot do that, it's going to be very difficult for the onslaught of long-read data that is coming. So one of the things that I've tasked our internal R&D group to do is to develop a scalable, efficient joint isoform caller. And a preliminary testing on over 100 samples shows you that it is very scalable. We can actually profile the mapped isoform of 225 million reads in 30 minutes, and that takes 55 megabytes of memory, and we can merge the isoform profiles of all of these in 15 minutes CPU time. And then to finally call the isoform GTF using 32 threads, we can do that in 20 minutes. We have to be able to jointly call and analyze isoforms on hundreds and thousands of samples, because that is what I believe is coming. Finally, just to show you, we also did some visualization. As you can see, we can generate very beautiful, nice full-length isoforms. I'm going to skip these. It's okay. I talk too much. But just to summarize, we have products for making long read, lots of cDNA reads. We have a large data set that I just showed you. It's over 500 million reads. And I really do think the future is scalable, efficient analysis of long-read RNA-seq data. And if you want to learn more, lots of stuff here. And you can always find me on Blue Sky, Twitter, and LinkedIn. Thank you.
Any pressing question that cannot wait? Very short, right? Because we are a little bit over time.
So I think most of the biologists care about the price, So, yeah, I'm developing methods for long reads, but actually, most of them for the ONT, yeah. So how much is this, the very high throughput PacBio cost? Yeah.
Come talk to me later.
Thank you again.